When you stop and think about nature, really, it's astonishing and amazing. And when thinking about microbiome data, it's almost more amazing because it's hidden. Bacteria are absolutely everywhere and on everything and are so important, but yet we don't see them. That's why I think that's really amazing to investigate, because it's this hidden, unseen side of science and nature, but it's really important. So my name's Lorna Richardson, and I'm a coordinator for the Microbiome Resources at EMBL Hinxton, which is a European Bioinformatics Institute. So EMBL-EBI is the home for big data. We collaborate with researchers, scientists and engineers all across the world to provide the infrastructure needed to share data freely and to make biomedical resources available. So I'm interested in microbiome data, so I look after the Magnify resource, which is EMBL-EBI's microbiome data and analysis resource. And I manage a team who ensure that the analysis that we provide is relevant to the field and that the way we present our results is really useful for users. A microbiome is the community of microbes or organisms that live within a particular habitat or biome. So most people think about human gut when we talk about microbiome, but actually anything that you can take a sample of has a microbiome. Any environmental sample or any sort of surface or even air all has a microbiome. There's actually lots of really good practical reasons to study microbiomes. For example, in medicine, you might be looking at the human gut microbiome and how that relates to health and disease. So you can compare which microbes are present within someone who's healthy versus someone who's sick. So usually what you're looking at is, number one, what is in there, so looking at which species of organisms are part of that microbiome. But then also we drill down into what those species can do, so which proteins they produce and what are the function of those proteins. A lot of the time what people are doing now with this microbiome data is they're mining it and they're looking for useful enzymes or useful proteins that might be helpful to us. So for example, they might be able to degrade plastics or harmful chemicals or improve efficiency of food production in agriculture. And to do that, you're looking for a very specific enzyme within the proteins of the database. So what we do is actually we generate pipelines. So a pipeline is a series of tools that basically do lots of different bits of analysis on the data. So we generate these very consistent pipelines because they have to be used for all different data types from all different places. And what that means is you can take the results of those analysis from different data sets and actually do some comparisons between them. And what that lets you do is really make use of this huge wealth of data that already exists in the public resources, so you don't have to always just generate your own data. It's super exciting to think that this is real world stuff we're looking at, because there's just loads of unknown out there. When we're studying microbiomes, some of the very well characterised ones, we're now getting a good sense of what's in there. But for the environmental samples, there's such huge diversity that we're always finding novelty. There's a lot of unknowns in there, so that's really exciting. We might find amazing stuff. These biomedical resources, the kind of resources that are provided by EMBL-EBI, are really crucial to all research nowadays. And so it's really nice to know that the work I do and what we generate is always going to be useful.